So as I mentioned, we're going to be starting in on the reproductive system. And the way I've decided to organize this information is we're going to go through some basic kind of concepts and characteristics associated with reproduction, talk about the anatomical structures that are involved with reproduction, and then we're going to hammer mitosis and meiosis pretty hard today. And then tomorrow we'll delve into the female reproductive system, which is kind of a few more moving parts than the male reproductive system does. So as far as definitions go, here are some definitions that you need to be familiar with. So gonads are, that's the scientific word that we use to describe the sex organs. For females, the gonads are ovaries, and for males, they are testes. And the function of the gonads is to produce what we call gametes. So gametes are for females, they are what we kind of the layman term we use for gametes for females are eggs. They are technically called ovum. And then for males, the gametes are sperm. Characteristics of gametes, one very important characteristic of a gamete is that it is what we call a haploid cell. So all of the cells, and a lot of this is just information that we're going to revisit that we discussed in 227, but all of the cells in our body, excluding the gametes, are what we call diploid cells. Diploid cells meaning that we have two copies of an identical set of what we call chromosomes, right? So we have 23 different chromosomes. The 23rd chromosome is the sex chromosome. And diploid tells you that there are two copies of each one of these 23 chromosomes for a grand total of 46 chromosomes per cell. The gametes, the eggs and the sperm, are haploid, meaning that they just have one set of those chromosomes. So they have 23 chromosomes. Another important characteristic of gametes is that 23 set of chromosomes that they have are genetically unique, right? There are subtle genetic differences between each and every one of the sperm that you produce if you're male and each and every one of the ovum that you produce if you're female. Genetically unique from the other cells in your body and genetically unique from one another. And then when gametes come together from genetically opposite sexes, so if you have a sperm that is able to fertilize an egg, you have those two haploid cells coming together, fusing to create a diploid cell. And because each one of those gametes was genetically unique, the cell that they make when they fuse, of course, is going to be genetically unique as well. So in addition to the gonads, the testes and the ovaries, we have accessory reproductive organs. And those accessory reproductive organs are there essentially to support the gonads and to help them to make viable and functional gametes. And then kind of the last part of this, last piece of this puzzle, and it's a very important piece, are the hormone profiles associated with reproduction. So we'll pretty extensively go through those, especially when we get to female reproduction, because female reproduction is cyclical, post-pubescently. The hormones are key to controlling those cycles. And again, we'll kind of get into the nitty gritty of all of that when we get to female reproduction. Okay, so the sex hormones, again, play a very important role, specifically in female reproduction in controlling the cycles. In male reproduction, the sex hormones, of course, also play a really important role in just allowing for sperm production, right? For males, just a kind of a prelude of what's to come. Testosterone is the male hormone that controls both female and male libidos, for that matter, which is the sex drive, but is also responsible for controlling sperm production. So when testosterone levels get too low for males, sperm production, the sperm that is made is oftentimes not viable. And we'll get into reasons why in a moment. Okay, so homologous structures. So if and when you decide to take developmental biology, you will talk a lot about homologous structures, right? And if you've taken 190, 192, you've talked about homologs between different species. There are also homologs within species. So what a homolog is it's a structure with a similar developmental origin, okay? So when we talk about this in the context of male and female reproduction, the homologue for the ovary, right, would be the testicle or the testes, and that makes sense. Um, the homologue for the clitoris in a female would be what we call the glands of the penis. So both of those structures, which are made up of sensory nerve endings, they are from a developmental standpoint, they have the same origin. The labia majora is homologous with the scrotum in the male. And then you can see there's a gland called the greater vestibular gland in females that is homologous with the bulbourethral gland, also called Cowper's gland in males. So I do expect you to know from this table these different homologs. Again, if you go on and you take developmental biology, you took a lot about homologous structures. Okay, so as promised, what we're going to really focus on today are kind of the steps leading up to puberty, right? The hormones that are involved in puberty, and then the production of the gametes, specifically with a focus on meiosis. And in order to understand meiosis, you really have to have a good grasp on mitosis. So we'll do a little review of mitosis too. So the word puberty is used to describe the onset of adolescence, right? At puberty, what happens is we get these hormone profiles, this change in hormone profiles in males and genetic males and genetic females that is different from what we saw in childhood, right? So once puberty begins and these hormone profiles come into effect, this is when we get the maturation and the production of viable gametes. So for females, females are born in their ovaries with all of the ovum, i.e. eggs, that will ever mature, that they will ever ovulate, right? So after puberty for females, every month you get this maturation process and then this event called ovulation, in which one of the ovum is released into the duct system for female reproduction. And then if the sperm is present, fertilization takes place. For males, males make sperm on a regular basis post-pubescently. So unlike females, males are constantly producing sperm, and the sperm that is not the viable sperm post-pubescently that is not ejaculated is reabsorbed by the male body. So the hormones. So this is the hormone profile that you find for both males and for females that sets everything into motion, right? So it all starts with a hormone called gonadotropin releasing hormone, GnRH. And GnRH, it's a releasing hormone, so that automatically tells you that this is a hormone. Pardon me, I'm a little bit stuffy. I think it's spring allergies. This is a hormone that comes from the hypothalamus, right? All of the releasing hormones come from the hypothalamus. So at puberty, the onset of puberty is marked by this increase in GnRH. And GnRH then travels to the anterior pituitary gland. And the anterior, this is again, just kind of by way of reminder, the anterior and the posterior pituitary gland have very different relationships with the hypothalamus, right? So the anterior lobe of the pituitary gland is connected to the hypothalamus through endocrine. It's an endocrine type of connection and communication, meaning that when the hypothalamus secretes a releasing hormone, it travels in the blood through a portal system to the anterior pituitary gland and then stimulates the anterior pituitary gland to release its specific hormone, depending on what was secreted by the hypothalamus. That's very different from the posterior pituitary gland, which is directly connected to the hypothalamus. So the hypothalamus, that's how the hypothalamus is able to make the hormones ADH and oxytocin, and then essentially makes them and then stores and secretes them in the posterior pituitary gland. Okay, but all the same, going back to GnRH. So GnRH at puberty is made and secreted by the hypothalamus, and then it gets into the blood and travels to the anterior pituitary gland, where it then stimulates the anterior pituitary gland to secrete two very important hormones. And those hormones are follicle stimulating hormone. The acronym of which is FSH. And so I will often just kind of default to using FSH, and another hormone called luteinizing hormone. Both of these hormones, hold on, I'll make this. Sorry, let me pause this. Okay, I'm sorry. Hopefully that was a bit of comic relief for everybody. I just was very stuffy. All right, so FSH and LH are important for both male and female gamete production, right? Deficiencies in either of these, if you're talking about males, would result in problems with male fertility. If you're talking about females, will result in problems with female fertility. Okay, the way these hormones work, oh, sorry, I had another slide for this. The way these hormones work, we'll get into kind of and fine detail when we go through both male and female reproduction. But in general, what happens is as you get an increase in follicle stimulating hormone and luteinizing hormone, these hormones will cause, in females, will cause the maturation of the follicles in the ovaries. And follicles are the structures within ovaries that contain the ovum, right? They are also the structures that secrete estrogen and progesterone. So as you get an increase in the size of the follicles containing the ovum and the ovaries, you're going to get an increase in estrogen and progesterone in females. And for males, both FSH and luteinizing hormone are required to essentially support support the maturation of sperm, right? And again, we'll kind of go into the nitty gritty details of that when we get to the specific in both males and females when we get to those slides. I guess the last kind of overview thing I want you to know is the FSH and LH are controlled by negative feedback. And most, when we talk about the endocrine system, most hormones are controlled by negative feedback, kind of the exceptions being, oxytocin during childbirth and prolactin and oxytocin when it comes to to nursing, to milk production and milk release. But more on that to come along the way. Okay. Let's see if there's anything from this side that I didn't say. Yeah, just by way of reminder, females, as I mentioned, have all of the ovum that they will ever ovulate from birth, and then males make and then ejaculate sperm post-pubescently. About every 2 weeks, you get a new batch of sperm that are made. So you get about 100 million that are produced per day, and then those that are not ejaculated within that 2-week period are absorbed. Okay, so this is just some vernacular that, again, kind of, by way of reminder, that we talked about in 227. So diploid cells, as I mentioned, that's all of the cells in your body are diploid, meaning that they have 46 chromosomes. This is how that diploid, so ploidy, the N is what we call the ploidy number. So the 2 preceding the N tells you that we are diploid, right? And then if you were to divide for the ploidy number N, you would get 23. So for humans, N is 23, meaning that we have 23 unique chromosomes. The 2 tells you that we have 2 copies of each. And then for the gametes, which are the egg and the sperm, those are haploid cells. So they're N23. And again, they are genetically unique, both from the cells of your body and also from one another. And then as far as other terminology is concerned, let me think. Yeah, I think that's pretty much everything I want you guys to know from that. Okay. The 23rd chromosome, as I mentioned, is the sex chromosome, right? So the skinny on the sex chromosome is that sex chromosomes can either be X or they can be Y, right? Anytime you have a Y chromosome, you are technically genetically male, right? So genetic males have 1 X, 1 Y. Genetic females have 2 X's. There are, of course, conditions in which some people have 3 copies of the sex chromosomes. So you could be profiles like XXY or XYY. There are also profiles in which 6 chromosomes can be missing. So, you know, X and then X0, what we call. So the take home right now for this class is if you have a Y, you're technically considered a genetic male. If you have an X and if you're the profile of XO, that's a condition called Turner syndrome, you're genetically female. So the Y confers specific male genetic characteristics. Okay, so now I'm just gonna do a quick kind of overview of mitosis. Because again, in order to understand meiosis, you have to have a really good handle on mitosis. So a couple things here. This is a great opportunity after we finish going through this. If you want to ask me any questions on mitosis and meiosis, I absolutely welcome you to do this. Some kind of helpful hints in revisiting this material and getting familiar with it is that it lends itself really well to drawing. So drawing pictures of what happens during each one of the phases of meiosis, I think is incredibly helpful. And this is also fantastic material to review during the LA sessions. Okay, and I'll run through it kind of quickly. And then if anybody, like I said, has any questions, feel free to ask me. So during mitosis, right? Okay, so during mitosis, what happens is we have a cell. Let's just take any cell in your body, a skin cell, which is going to be a diploid cell, right? And that diploid cell is going to have, again, 46 copies, so 23 chromosomes, 2 copies of each. And as we go, and actually, I'm going to skip through this slide so I can get to, nope, I'm going to have to draw it. Okay, you guys, bear with me. Pen. Okay, so this is, what I'm drawing right here is the cell cycle, okay? That's supposed to be a G. G1, G2. Oh, I'm sorry. What am I doing here? How do I erase? This is supposed to be S here. And G2. Okay. It's like a caveman drawing. All right. Okay, so all of this in here, this is the cell cycle, right? And all of this in here, G1, S, and G2, this is all what we call interphase, right? It's just that it's used, that word interphase is used to describe the life cycle of the cell, right? So a cell, as it's going about its business, is going to cycle through each one of these, right? So in G1, this is just a cell doing its thing, you know, making, if it's a liver cell, it's making its liver proteins. If it's a skin cell, it's, you know, making, if it's a skin cell stratified squamous epithelial cell, it's making keratin, just again, kind of going through its business. Then it enters, during interphase, it enters this phase called S, and S stands for synthesis, right? So during the S phase, what happens is that for this cell, each one of its 46 chromosomes, because it has 2 copies, right, of the 23, each one of them undergoes synthesis and makes a duplicate of itself. And that duplicate is stuck to it, right? So that is what you're seeing in this picture here. This would be, let's say, chromosome 1. After this cell has gone through S, chromosome 1 is going to have a copy of it stuck to it by its centromere. And then these 2 arms are called sister chromatids, and they are genetically identical to one another. All right, during G2, the G, by the way, stands for gap, in case anybody is interested. So during the G2 phase, this is the phase during which The cell goes through to checks to make sure that during the duplication process, no major mistakes were made that couldn't be fixed. If mistakes were made that can be fixed, then that's great. Then the cell fixes those mistakes. If there are mistakes that are made that cannot be fixed, then at the end of G2, that cell is prompted to go through what we call apoptosis, which is pre-programmed cell death. And again, we're doing an overview of meiosis and mitosis, which is going to be the same for meiosis. And then if everything looks good, the cell will then, now that it has, because again, it has twice the amount of genetic information, it will go through, in this case, I'm going to describe it in terms of mitosis, right? And then here are the 4 phases of mitosis, which are the same 4 phases of meiosis. The first one is prophase, right? I gave you guys that little kind of mnemonic device, pass my anatomy test, as a way to remember the 4 different phases. So the first one is prophase. Hallmarks of prophase is that during this, and again, the whole point of this process is to take this now, because the cell has duplicated its genetic information. So now the entire point of mitosis and even meiosis is to separate out this duplicated information. So during prophase, what will happen is you'll get dissolution of the nuclear envelope, and you'll get condensation of the chromatin, because that's the way DNA is found when the cell is in interphase, is in chromatin form. So you'll get condensation of that chromatin into chromosome form, which is what we're seeing here. All the cartoon pictures that are drawn when we talk about mitosis and meiosis are always shown in chromosome form. And so by the end of prophase, you have the DNA is roughly looks like this. I should point here, roughly looks like this. It's pretty condensed into chromosome form. During metaphase, you get the lining up. That's what you're seeing here. You get the lining up of those chromosomes on the metaphase plate in preparation for separation, right? You also get spindle formation. So you get spindles that come from the centromeres that reach out. They're kind of like fishing line, reach out and attach to the centromere of the chromosomes in order to pull those chromosomes apart, right? Because again, if you think about this chromosome has 2, you can see it's got 4 arms. So these 2 arms right here are they're just identical genetic copies of these 2 arms. You only want functional chromosomes look like this, right? This is not a functional chromosome. This is one chromosome that's stuck to its sister chromatid. I mean, we call both of these sister chromatids because they're, again, genetic copies, identical copies. So all the same, so they line up against on the metaphase plate in preparation for separation, and the spindle attaches to the centromere. And then during anaphase, what happens is you get constriction of the spindle, and so the centromeres start to contract, and this pulls apart the centromere and it pulls sister chromatids in opposite directions. So you get this sister chromatid being pulled toward this pole, and this one being pulled toward the opposite pole. And then finally, the cell moves into what we call telophase, and telophase happens after all of the sister chromatids have migrated to the opposite poles of the cell. During telophase is kind of the reverse of prophase, with some important differences, one of which is that during telophase, you have 2 cells that are stuck to one, they're still connected to one another. So they're not separate cells yet, but during telophase, you get reformation of the nuclear envelope, you get relaxation of the chromosomes back into chromatin form. And one of the hallmarks of telophase is you have what's called the cleavage furrow, if you can remember that. And what the cleavage furrow looks like, just to draw a very kind of basic elementary picture of the cleavage furrow. This is the cleavage furrow right here. So this would be a cell in telophase that is not yet 2 separate cells. Kind of draw the reformation here of the nuclear envelope. This is what it looks like under the microscope. Feel free to revisit this chapter. And then you get kind of this, will look like scribbles within the newly forming nucleus or nuclei as those chromosomes start to relax back into chromatin form. So it's going to be two again, that it's one cell that is almost two. And then the last part of telophase is a separate, I shouldn't say it's the last part of telophase. After telophase is complete, there's one last event called cytokinesis, and that is the pinching off of the plasma membrane so that at the end of cytokinesis, you have two separate cells ready to go right back into gap one. Okay, so like I said, that was a really brief overview of mitosis, really, so that we can go in and talk about the nuances of meiosis. Okay. So how does meiosis different than mitosis? Meiosis, you can think of meiosis. I think an easy way to think of meiosis is, first of all, keeping the overall goal of meiosis in mind. So at the end of mitosis, we have one cell that duplicated its genetic material and divided out to make two cells, both of which are diploid and genetically identical to one another. During meiosis is the cell going through that kind of like through a mitosis type process twice, right? So we call meiosis, there's going to be a phase 1 of meiosis and a phase 2, okay? And so we're going to have prophase 1, metaphase 1, anaphase 1, telophase 1, and then we're going to have prophase 2, metaphase 2, anaphase 2, telophase 2. So the first phase of meiosis, the goal of that phase is to scramble up the genetic information within the cells that are going to divide out to become the gametes. And then the second is the second phase of meiosis is essentially just mitosis. Okay. And that'll make sense here in a moment as we go through the different phases. Okay. So we're first going to go through meiosis, all of the phases in meiosis 1. Okay. And again, as far as the hallmarks of each one of these phases are concerned, they're the same as what you would expect to see for prophase, metaphase, anaphase, and telophase. So during, one other kind of piece of background information that is important to keep in mind to help you understand this process. The only cells that go through meiosis are the stem cells for the gametes, okay? So what I mean by that is the oogonium, or the spermatogonium. Those are the cells that are going through this process to make the gametes. Okay, the gametes themselves, and I think that's a common misconception, or people just don't understand that when they're first trying to wrap their brains around meiosis. This is the process that makes sperm and or makes eggs. The eggs and the sperm themselves don't go through this process, right? It's the stem cells for those that result in the production of the functional gametes that go through meiosis. So during prophase 1, what happens during prophase 1, just like what we see during normal prophase, is you get let's just use the spermatogonium in this case as an example. You get the spermatogonium going, you're getting dissolution of the nuclear envelope, and you're getting condensation of the chromatin into chromosome form. Okay. And then something very special happens during prophase 1 of meiosis. What happens is that, so let's just say that this is, you have chromosome 1 and chromosome 1 from mom and chromosome 1 from dad, right? And so here you can see this is the duplicated arm of this one, and this would be the duplicated arm of this one. So for chromosome 1, those chromosomes saddle up next to one another, and they form what's called, they undergo what's called synapsis. Okay, and some more terminology. When you have chromosome 1 from, I'll draw my own, right? So here is chromosome 1, and its duplicated arm, its sister chromatid, that you got from dad, okay? And I'll put a 1 here. And here's chromosome 1 that you got from mom with its sister chromatid. So during prophase 1, these chromosomes, and just a little bit more terminology, when you have an intact centromere, we call it a chromosome. So even though these are sister chromatids, because they are connected by an intact centromere, we just call this structure a chromosome. So chromosome 1 from dad during prophase 1 of meiosis saddles up next to chromosome 1 that you got from mom, and you get this, they form what's called a tetrad, right? And they undergo what's called synapsis. And a synapsis is a connection, right? So you can see it well here. So they saddle up next to one another. They synapse, they form a synapsis with one another. And then you get something called crossing over. Okay, and crossing over is the event during which, for a particular allele, right? And remember, an allele is a gene that codes for the same phenotypic trait in one chromosome versus another. So if we're talking about the trait is eye color, and let's just pretend, although this is not true, that eye color is just controlled by one allele, okay? What happens during synapsis is you get the formation of this tetrad, and the information, the eye color, the allele that's on dad's chromosome trades places with the allele for eye color on mom's chromosome. So they swap genetic information. They start to scramble up the genetic information during prophase. And that's what you're seeing in this picture here. That's why you're seeing, if this is a chromosome 1 that you got from dad, you can see here that at the end of prophase 1, this chromosome now has a piece of what was originally the maternal, that has the maternal allele swapped out, right? These alleles swapped places. And that's really, that's a very important concept when it comes to the result of meiosis, which is the production of genetically unique gametes. Okay. So during metaphase 1, again, it's metaphase, so you're getting the chromosomes lining up on the equatorial plate. Because we're talking about meiosis, what you will get is you will get moms. So these, you can't see it in this. Oh, you can see it in this one though. So you can see how here are this scrambled up now chromosomes. This was original dad, this was original mom, and they line up on the metaphase plate, this equatorial plate, and they line up side by side, right? And that's another kind of important distinguishing characteristic to keep in mind when you're drawing out your own phases of meiosis. They line up side by side, and the way that they line up side by side is there's something called random assortment, okay? And what random assortment means is that for this particular cell undergoing meiosis, you might have, because here, these are the opposite poles of the cells, right? So for this particular cell, you had what was original chromosome 1, paternal chromosome 1 is on the left, right? And original maternal chromosome 1 is on the right. For chromosome 2, on the left, you have the original maternal chromosome 2 on the left, and the original paternal chromosome 2 on the right. And then for chromosome 3, in this particular picture, you have paternal on the left, maternal on the right. For another cell, the way that these cells line up is completely random, right? You don't know whether or not it's going to be maternal or paternal chromosomes on the left versus the right. And there are some really important implications for this when it comes to genetic diversity, because during the next phase, anaphase, where you're going to get splitting of the, or not, in this case, actually not splitting the centromere, but you're going to get movement of the chromosomes to opposite poles. For one cell that has, let's say, like in this picture, paternal on the left here, maternal, paternal for chromosomes 1, 2, and 3, another cell might have all paternal on the left, right? So when those chromosomes move to opposite poles of the cells, they're going to be genetically unique from one another, just insofar as which original chromosome they had that's moving to a particular pole, north or south, if you will. Okay. And And then during anaphase, so an important distinguishing characteristic of anaphase 1 in meiosis is that during anaphase 1 of meiosis, the centromere does not split. Okay, so make a little asterisk, a little note to yourself. The centromere does not split. So the entire chromosome gets pulled over to either pole of the cell. Okay, such that at the end of telophase 1, which telophase 1 is pretty much just telophase, you get the development of the cleavage furrow, and then cytokinesis will take place. You're going to have, after telophase 1 is completed, you're going to have two separate cells. And here again, another asterisk to yourself, two separate cells that are haploid cells. Okay, they're haploid because they only have one copy of 23 chromosomes, right? But those haploid, those haploid chromosomes are not going to be functional because they're still attached to their sister chromatid. All right, and that is because the centromere did not split in anaphase 1. Okay, so as soon as telophase 1 is cytokinesis is complete, those cells, those pre-gametes, immediately go in to meiosis 2. Okay, and meiosis 2 is basically mitosis from when you think about the way in which it occurs. So during meiosis 2, what happens is, again, you're going to get dissolution, because at this point you need, now the chromosomes have to kind of recondense, but they, because of recondensation of the chromosomes, you get lining up of the chromosomes on now, which are attached to their sister chromatids on the metaphase plate. During anaphase 2, so during anaphase of meiosis 2, the centromeres do split, okay? And so what you get at the end of meiosis 2, instead of the production, because you've had the cell go through this process twice, you're going to have 4 cells instead of 2, which is what we see in mitosis. And each one of those cells is going to have half of the amount of genetic information because of the splitting of the centromere in anaphase 2. Okay, so we call meiosis 2, we call it the reduction division, because you're going from the whole point, if you will, of meiosis 2 is splitting that centromere and dividing up those sister chromatids into 2 different cells. And remember, those sister chromatids after meiosis 1, they're genetically unique from one another. So when at the end of the day, when you get the production of these 4 haploid cells, each one of those haploid cells is going to be genetically unique from each other. Okay, so let me see. So here I like these pictures, and they actually conveniently come from your textbook. The reason I like them is because it reminds you how during meiosis you get the scrambling of the chromosomes, right? So at the end of the day, after telophase 2 and cytokinesis has occurred, you're going to have a cell that has it's a haploid cell, so it has all 23 chromosomes, but just one copy of each because of the splitting event that occurred during anaphase 2. And each one of those chromosomes is genetically unique from one another. It's also a mixture of, you're going to have, because of random assortment that occurred during metaphase 1, you're going to have some cells that are produced that might have what was originally mostly maternal chromosomes, but with bits of paternal DNA stuck in them. You might have the production of some gametes that have kind of a even split of original maternal and original paternal, but that are mixed up. You might have some cells that are made that are mostly original paternal, but with bits of maternal stuck in there. Does that make sense, everybody? So it's really the random assortment during metaphase 1. Of course, all of the events during prophase 1 with regard to the synapsis, the formation of the tetrads because of the synaptic event and crossing over that occurs. All of those result in this highly diverse from a genetic standpoint population of cells that we get when it comes to gamete formation. Okay, does anybody have any questions? Okay, whoops, sorry. I think I'm gonna